Welcome to Future Talk. On today's program, we're going to talk about genomics and how genomic science is leading us to a new world of personalized medicine where your genes can help predict the likelihood of certain diseases and indicate specific treatments tailored to your personal genetic makeup. My guest is Michael Snyder, one of the world's top genetic scientists. He is the Stanford Wasserman Professor of Genetics, Chairman of the Genetics Department, and Director of the Center for Genomics and Personalized Medicine at Stanford University. He's also a co-founder of several successful biotechnology companies, a pioneer in the use of big data in medicine, and the author of the recent book, Genomics and Personalized Medicine, What Everyone Needs to Know. Mike, great to have you on the program.
Thanks for having me.
We're going to get into your work in just a couple of minutes, but first we have a short animated video that gives some of the basics of genetics, put together by a group called Speaking Clearly. We've edited it slightly for length. I think you'll enjoy it. Let's go ahead and roll that video.
Stated Clearly presents, what exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckle gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. I'm John Perry, and that's Genes Stated Clearly.
So that was a brief introduction to genes. Now I'm interested in your book, Genomics and Personalized Medicine. You have a copy of it right here. Maybe you could hold it up to that camera so we can see it. Well, let me ask you this. How does knowledge of your genome aid in your health? How does it enable people to provide personalized information about your treatment?
Okay, well these days your genome information is used in 4 different areas of medicine. One is it's being used to treat cancer. Another is to solve mystery diseases. People have diseases they haven't been able to figure out. They're getting their genome sequenced to try and understand what's wrong, mostly in children. A third area is actually in prenatal testing, so trying to understand if there's a genetic defect in a child before it's born. And lastly, in risk prediction, that is trying to predict the diseases you might get. If you're healthy, your genome information can be used to predict diseases that you're at risk for.
Now, can you look at somebody's genome and say, if you have this gene, you have an increased likelihood of getting cancer, let's say? Can you pin that down to maybe 1 or 2 genes?
You can. It's more than 1 or 2 genes. Famous examples, for example, are BRCA mutations. So what's done is if you've looked at these genes called BRCA, if they have certain mutations in them, you can be at high risk for breast cancer or ovarian cancer if you're a woman. So mutations in those genes are very predictive of breast and ovarian cancer. And there's the famous case of Angelina Jolie, who actually had familial breast cancer and got tested and discovered she had a mutation in one of these genes, and then actually took action, had her breasts and ovaries removed.
When you say a mutation, does that mean that something bad happened to a gene that broke it or rendered it dysfunctional?
It does, yeah. So what happened in her case was inherited in her family. So it was running in her family, and she inherited a bad copy of this gene. And so the consequence was she knew that put her at very high risk for breast and ovarian cancer.
What makes a gene go bad? Does it like bump into something that makes it break?
Yeah, sometimes it's spontaneous mutations that just as DNA replicates, there can be, or it makes copies of itself. There can be errors that will come up as it does that. They're not frequent, but it does happen. But there's also environmental situations that can cause this. So, for example, people who smoke are actually damaging their DNA, especially in their lungs. Another good example is people who are in the sun too long. The UV light can damage your DNA in your skin, put you very high risk for skin cancer.
Now, if you have 10 trillion cells or so in your body, and they all have the same genetic code, so if you damage the DNA in some of those cells, how big a difference does that make?
Well, if you damage the wrong genes, these cancer-causing genes, they can start growing out of control. So most of the cells, not all, but most of them have actually finished dividing, and they're sitting there, or they're dividing very slowly. And if they get damaged and make an error in a gene that causes them to grow out of control, they may start doing that, and that's what causes cancer, actually. It's cells growing out of control because of a genetic mutation.
Now, how does a cell know when to reproduce? Does it need permission to reproduce, or can it just say, I'm not taking orders anymore, I'm just going to reproduce as much as I want?
Yeah, well, some of your cells are generally pretty done dividing. Your skin cells do replace each other, your liver cell divides, but your pancreas, for example, and your kidney cells don't divide much anymore. Once you're done making your kidney, for the most part, it's done. There aren't lots of new cell divisions that occur. But certain areas, like in your gut, where you're actually digesting food, those cells replace themselves. They divide actually quite a bit. And your blood cells actually divide quite a bit. So certain cells are dividing all the time, and others not so much. Your brain cells don't divide very much, for example.
Now, is this field growing exponentially? There are certain branches of technology that are going at an exponential rate. Is this one of these fields where in 5 years from now we're going to be in a totally different environment?
Oh, I think that's already true. I mean, even 10 years ago, you couldn't imagine getting your genome sequencer. Maybe it was just on the edge. 10 years ago, there was probably maybe 1 or 2 genomes that were sequenced. Actually, take that back. There's only one, what's called a reference genome, that was sequenced. So just a theoretical copy, so to speak. The first personal genomes, first people getting their genome sequenced, that happened in around 2008. And then now it's amazing. There's over there's over 1 million genome sequences that are out there. It's not commonplace for everyone yet, but it could be in the future. And so 5 years from now, getting back to your question, I could envision a world where people will be getting born with their genomes already sequenced.
So being sequenced in utero, wouldn't it be easier to wait until they were actually born? Or maybe while they're in the womb, you could see that the genome was defective. Maybe you choose to terminate the pregnancy at that point?
Yeah, that's part of this new prenatal diagnostics they talk about. So that is to say, in the old days when you had tests, for example, people are at high risk for Down syndrome. The older mothers would be at high risk for getting Down syndrome, which is an extra copy of one of your chromosomes. It's chromosome 21. And that was screened by a very laborious, actually, semi-dangerous procedure. That is to say, you'd go with a needle and you'd actually poke into the womb, pull out cells, and try and see what was wrong. Now you can do it actually just by drawing blood from the pregnant mother. It turns out the fetus, that's growing in the womb, actually sheds a little bit of DNA into the mother's blood. And you can actually draw the mother's blood, and then by sequencing it, you can actually determine, these days it's mostly just looking for chromosome changes, not down to individual letter changes, but chromosome changes. You can tell Down's children and actually some other chromosome changes just by sequencing the mother's blood. And in the future, people will actually be getting entire genome sequence before the child's born.
Now, I understand that you had your own genome sequenced and you found out some stuff that caused you to make a few changes. What was that all about?
Yeah, so I was one of the early adopters of this. I sequenced my DNA very early on. I'm probably one of the top 20 people to get their genome sequenced. And we looked at my DNA to see what diseases I might be at risk for. And what was discovered is that a lot of things I already knew from my family history. I'm at high risk for heart disease. I knew that because of my father's side. They've died of heart failure and such. At low risk for obesity. Nobody in my family's ever been overweight. But one surprise was that I was at very high risk for type 2 diabetes, which I was not aware of from my family history. And we had been making many other measurements as well, besides just looking at my DNA, my genome. And what we discovered was that as we were following along, suddenly my glucose spiked through the roof, which is what happens when you get diabetes. Your glucose goes out of control. And so I was on the alert for it because my genome sequence said, I am at high risk for type 2 diabetes. And then because I was following then my glucose levels, I discovered when it first went off. And it went off to the point where I was classified as diabetic.
Do we expect that sometime in the near future, it'll be routine, like every baby or even every infant in the womb will be sequenced, and then by the time they're born, we already have a genetic blueprint of them, and we can use that for various purposes?
I think there's a very good chance that'll happen. This is still very controversial right now. It's even controversial whether you should be sequencing people while they're healthy. At least for some people, that's controversial. It's not for me. My own view is that I think it will become standard in the future that people will get their genomes to better manage their health. Once you know what you're at risk for example, much like if you knew you were at high risk for breast cancer. And by the way, men who have mutations in this BRCA gene, this breast cancer gene, actually become high risk for breast cancer as well. What will happen is you'll get screened for that more. So this information can be very, very valuable for managing your health.
Is there any reasons why a person would not want to be sequenced? Like maybe they want to keep it private. Is your genome your property or is it public information?
Oh, great question. There's two reasons why people might not want to get their DNA sequenced. They may not want to know. They don't. For example, some people are very severe worriers. That is to say, if you knew that because you sequence your DNA, you're at high risk for breast cancer, and every morning you got scared of getting breast cancer when you woke up, that would not be a healthy situation either. So getting scared is one reason why people don't do it, or I should say just worrying about your risks. And that's a perfectly justifiable reason why you might not want to do it. The other reason is the one you just said, privacy, that a lot of people are concerned about their genetic information getting out there and somehow being used against them. People are worried about that. I'm not aware of any significant cases of that happening, but that is a concern people have.
Maybe it would be harder to get a health insurance if you had a risky genome.
Yeah, believe it or not, there are actually laws to prevent certain kinds of genetic discrimination, it's called. So you're not allowed to be discriminated for employment or for health insurance based on your genome information. But you can still be discriminated for life insurance and long-term disability. So that is a possibility. Now, my own view is it's still better to know what diseases you're at risk for and have your life insurance go up, because it's better to understand what you're at risk for and therefore try and take steps that might avoid getting those diseases.
Now, I understand that your genome can not only predict what you're at risk for, but it can recommend specific treatments like certain types of cancers. Your genome can indicate what type of treatments might work for you.
That's absolutely correct. And so cancer is probably the biggest field where genome information is having impact. So cancer is a genetic disease. It takes genetic mutations. You can either inherit them, like I mentioned earlier, but also as you go along in life, you do accumulate mutations. And if you get some bad mutations in genes that cause cells to grow out of control, the cells will start growing out of control, basically, and start forming tumors and cancer. And so because it's a genetic change, it's turned out that sequencing people's DNA has been very, very valuable for treatment. And just as an example, in lung cancer, what's very clear, actually in all cancers, that the actual mutations varies from person to person. So if you get cancer, I can't know for sure what the genetic change is that's wrong with you. But if I go in and sequence your DNA, I will find that change, and sometimes that suggests treatments. So going to lung cancer, it turns out that there are 5 different genetic pathways that get mutated, probably more, but at least 5 in lung cancer. And depending which pathway is mutated determines which therapy you'll get. So if you have a mutation in a certain kind of pathway, you'll get one kind of treatment. And if you have mutation in another kind of pathway, you'll get a different treatment. And you really need this information, otherwise you'll get the wrong drug for the wrong condition.
Or maybe everybody will get the same treatment and it will only work for some of them.
Yeah.
Because some of these drugs have devastating side effects, so you don't want to take the wrong drug.
That's absolutely right. Yeah, so this is what precision and personalized medicine is all about. It's a matter of getting the right drug for the right person at the right time.
Now, reading the genome is one thing. Another possibility is writing the genome, in other words, changing the genes. Is there work being done to try to modify your genome so you're not susceptible to certain ailments?
The answer is yes. It's not really so much for susceptibility. It's being done much more for treating conditions. So one area that's being tested and now tried is actually, first of all, there are tools to go in and change your DNA that are fairly specific, that can make changes. And it's still not clear how often they'll have side effects. But if you're in a bad situation, having this therapy can be useful. And one area where it's being tried is actually for AIDS patients. It turns out if you can mutate what's called the receptor, the protein that the AIDS virus binds to, the thought is that it won't infect those cells as well. And so actually for someone who has AIDS, it's being tested that to remove this protein that the virus binds to should help those patients. That's being tested. And there's a few cases like that as well.
Now, you're considered the pioneer in the application of big data to medicine. And for people who don't know, big data is a computer term where you analyze huge quantities of information in order to detect patterns that would not be visible otherwise. So how would that apply to personalized medicine?
Yeah, so your genome is just one part of this. It's a lot of information in your genome. You have 6 billion letters that basically program you, so to speak. But it's only, believe it or not, still a very small part of all the information that's useful for managing your health. There's an area called epigenetics and epigenomics where there are changes that can occur on your DNA that can be useful for managing your health. There's other kinds of information as well. Making molecular measurements of other sorts is powerful. It turns out that wearing wearable devices, so smartwatches and things, we've shown and now other groups have shown can be very valuable for managing your health, we believe.
So is it good to constantly be aware of every change in your heartbeat or your respiration? Do we want to monitor ourselves and keep ourselves under surveillance all the time? Is that going to make us healthier? Or is there such a thing as being too worried about our health?
That's a great question. And the answer is, so that's a personal choice, so to speak. My own view is that you can't have enough information, but you don't necessarily want it hitting you in the face all the time. My own view is that this should be much like a car dashboard. So whether you know it or not, you have 400 sensors on your car, and it's always measuring things. But your dashboard only has a few lights, and when something's not quite right, those lights go off. That's how I see personalized and precision medicine of the future. You'll have all these things that you'll be doing either in the doctor's office or with wearable devices. And they'll be constantly measuring you, but you won't know it. You won't worry about it. But what will happen is when something's not quite right, one of these lights might go off and say, well, maybe you better stay home today, or maybe you better go visit a doctor.
Now, science has discovered a lot of things relating to health recently in your body. There's something called the microbiome, which is a set of cells inside your body that don't really belong to you. They're foreigners, but they serve a vital role. How does this tie in with personalized medicine?
Yeah, believe it or not, we have anywhere from 2 to 10 times as many cells in our gut and on top of us that are not us. They're mostly bacteria, but there's also fungi, amoeba, all kinds of microbes live in and on us. And that sounds really scary, I know. And people hear about microbes in a nasty way, but most of these microbes are actually absolutely essential for your health, and they're beneficial for you. So your gut bacteria, for example, that digest your food. You cannot digest most of your food without these microbes. You need your gut, you need your bacteria to do that.
Now, how do they get there? Presumably the womb is kind of a sterile environment. There shouldn't be too many foreign organisms in there. So do you just pick up all these organisms through living in this environment, and they just kind of take up residence in you and then turn out to be useful?
Well, you actually get the first part of this from your mother, but let me back up a little bit. The other thing, in addition to digesting your food, it'll actually make essential vitamins. So your bacteria don't just help break down, they actually make things that are very essential for you. So if you have ever vitamin B12, many other vitamins are only made because of your bacteria. So you really do need these things. They're very, very important for your health. But where do you get your microbiome from? Well, you get it at birth. So we're born relatively sterile, so it's thought. But actually, the very first thing that happens is you come out of the womb. And the microbiome from so these bacteria that you first get actually come from the mother's vaginal region. That is to say, you pick up microbes, and that actually will show up on the skin and other places. And so babies that are born by vaginal birth will have a different microbiome than those born by cesarean. The cesarean-born children tend to look more like the skin of their moms because the moms are holding the babies near the skin. So your microbe actually initially comes from your mom, and then from the food you eat. As you eat food, you're feeding your microbiome, so to speak, and it's growing and developing, and by age 3, it pretty much stabilizes.
It's interesting that it sort of gets there without any planning, sort of accidental, but it ends up playing a vital role. Like, what if it weren't there? Could your body do the job that your microbiome does?
No, I think you'd be pretty sick. It's pretty clear that mice, you can make mice, which are a great model organism for studying this sort of thing. If you make mice lacking a microbiome, they're pretty sick. And it's not even clear people would live if they didn't have a microbiome.
So I have an interesting question about genetics. Like, how does the cell know how to differentiate? There are tens of thousands of possible specialized cells that a stem cell could become. Is there some kind of consciousness or intelligence saying to each cell, you're going to be a skin cell, you're going to be a bone cell, you're going to be a muscle cell, so look up the gene that describes that and start taking that form?
Yeah, it's all programmed in our DNA. So as you know, we all form from a single cell. When a sperm fertilizes an egg, you have now the starting material, if you will, for a human being. You basically have a single cell, and that will grow and divide. And there's a very specific pathway, which we don't fully understand. It's a very active area of research and science. People are trying to understand, how can you make such a complicated human being with 10 trillion cells, with all these different organs and all these different cell types you mentioned. And there is again, it's programmed from your DNA, and it's not to say it can't be changed. It can be changed by the environment, but clearly your DNA has a very, very big impact. That's why twins look alike, right? Identical twins look very similar, although they're not 100% identical.
What's the biggest bottleneck in your research? I mean, aside from funding, obviously, what are some of the toughest technical problems to crack?
Well, there's a number of them. I would argue the way we sequence DNA now is still very slow and cumbersome, and we still miss certain kinds of mutations. So believe it or not, what we're very good at detecting when we try, for example, if I want to see the variants in my DNA and how do they compare with yours, we'll actually break the DNA into small bits, and then we'll sequence millions of those bits at once, if you will, to try and find changes. And it's good at finding little small changes, but it's not so good at finding big changes. And so there are newer technologies for being able to do that. They're more expensive, so we need to figure out how to do this so we can capture all the changes in a person. And the other is it takes too long. The fastest you could sequence a person now is, you'd sequence say the relevant DNA you can detect, it's roughly 24 hours. And even still, but the normal time is more like 2 weeks. And in some cases where this is life and death, where there are children that are born into neonatal situations, so preterm birth babies can often be very ill. And actually sequencing the DNA, it's thought will help a number of them. But they don't have much time in many cases. So you really want to get a sample of their DNA, sequence it as fast as possible. And if you do learn something useful, relay it back to them as quickly as possible.
Now, this might sound a little bit science fiction-y. We're talking about modifying genomes to make you more resistant to certain diseases. Do we see on the horizon modifying genomes to create different traits, make people bigger, stronger, faster, smarter, things like that, tailor-made people?
Yeah, I mean, this is obviously an area of intense interest and obviously one of high ethical issues, shall we say, that people will have to deal with. And right now, we don't have the information to be able to do that. That is to say, most of these traits you describe, like intelligence and things like that, first of all, measuring them is tricky. But second, they're probably not due to a single gene. That is to say, there's probably multiple genes in combination that make up those traits. And so we don't have that fully figured out, so that's not so easy to do right now. But at some point, it will become an issue in the future, and then the question is, how do we deal with this? You know, I don't think anybody will argue that changing the DNA to stop a nasty disease is a bad thing. If you can prevent a child from getting cystic fibrosis or Tay-Sachs disease, then no one will argue that's a bad change to make. But it becomes an issue when you're trying to make someone who should be if you had the information to make someone 6 inches taller, should you go around and doing that? And this is something we'll have to deal with. I mean, it's an issue already, as you know. In China, there are many more male babies born than female. And so clearly, that's not random. There is some selection going on, and those are ethical issues that groups have to deal with.
There are lots more questions I'd love to ask, but I've gotten the signal we're out of time, so we are going to have to wrap right now. I've been speaking with Michael Snyder. His book is called Genomics and Personalized Medicine. Thank you for watching. Visit our website, www.futuretalk.net. For Future Talk, I'm Marty Wasserman, and we'll see you next time.